Howdy folks, in this microbiology video we're going to be talking about the serial dilution that we will be using to actually isolate colonies on a spread plate. Now, this method is slightly different from what you will see in your textbook, in your lab notebook, so make sure that you are following these methods as these are what best fit our facilities, okay, with this number of students. Now, what you need for this method is you need a sterile pipette in a baggie, sealed baggie. You need scissors to open that up. You need a spreader, a beaker of isopropyl alcohol with a glass cover. You need a disposal container. You need the plate that you're going to spread onto, and then you need 3 tubes, one of which is your bacterial source tube, and the other 2 are sterile, fresh media broth tubes that you will be transferring this bacterial culture into in a serial manner. And then lastly, you need the Bunsen burner to help keep everything sterile, along with the inoculation loop for transfer. Now, for this method, before you get started, the easiest thing that you might do is go ahead and cut open your sterile pipette bag. Now, you don't want to cut it open too early, as soon as it's open you risk potential cross-contamination. So do that right before you start this. Get your inoculation loop from the tray on the side. Sterilize it, starting at the stem, then up to the loop. Let it cool. Go ahead and get your 2 tubes in hand. You want your source tube and tube A in hand. Okay? Now, once your loop has cooled enough, decap the source tube, flame it, retrieve bacteria on your inoculation loop, flame it, cap, decap tube A, flame, and inoculate. Flame, cap, sterilize. Now, you're going to do this again with the same tube. We are essentially going to inoculate tube A twice. Let this cool. And we're now going to do that second inoculation. So again, decap your source tube, flame it, reach your inoculation loop in, collect bacteria on the loop, recap, decap, flame, and inoculate, flame, cap. Now, you may have heard me bump the handle against the side of the tube, so I'm going to pass the handle over the flame a little bit there, and then sterilize. Now at this point, I'm done with my source tube. I'm going to set it aside. And now I'm going to get both tube A and tube B in hand. Okay. Let my inoculation loop cool a little bit. And now, the bacteria that we transferred from the source tube into tube A, we are now going to serial dilute and transfer some of that culture over into tube B. Now, with that, we want to make sure, because we just transferred it over, we want to make sure that this is evenly distributed. So, for the moment, hold your inoculation loop in your other hand and simply flick tube A to mix it around. Okay, to make sure you mix that bacteria around so that when you reach in to move it into tube B, you have it sufficiently mixed. So, decap, flame the tube, reach in to tube A, pull out, flame the tube, cap, decap tube B, flame, reach in to inoculate, flame the tube, cap, and sterilize. Let it cool, and do it again. For each of these transfers for this serial dilution, we are going to have 2 inoculations from the source tube into tube A, and 2 inoculations from the A tube into tube B. So once this is cooled again, remove the cap on tube A, flame, reach in to collect, swirl around a little bit, flame, cap, decap tube B, flame, inoculate tube B again, remove, flame, cap, and sterilize. Okay. Now, once this cools down, we're going to set this down because we're done with the inoculation loop at this point. So let it cool. Once your inoculation loop has cooled, you can set it aside because we're not coming back to it. Now with tube B, you want to make sure you flick it to mix it, alright, so that the bacteria that you have moved into this tube is evenly distributed, and set this down for the moment. We're now going to prepare the spreader for use on the plate. In order to sterilize the spreader, we're going to make use of the isopropyl alcohol in the beaker here. Now again, there's a glass plate here for a reason. We'll come back to that in just a minute. But for right now, simply take the glass plate off and set it aside. With your spreader, simply dip it in to the isopropyl alcohol and move it over to the flame. And let it flame. Keep it near the flame, alright? If for some reason a drop of flaming isopropanol finds its way into your isopropanol beaker, simply take the glass cover and set it on top. Fire requires oxygen, and by doing this you remove oxygen from the equation and you put the fire out. Don't freak out, don't go pull the fire alarm, just simply take the glass cover plate and set it on top. And then get back to what you were doing. Alright? So, the other thing I want to point out here, and this is a good time to address this, because I have a personal anecdote to share. It is very tempting to sit down and do all this work in the chair. But I have a graduate school friend who, when she was doing this process, she got her spreader soaked, flamed it, and turned around to talk. Okay? And while she was sitting, a drop of flaming isopropanol fell on her leg, which she was wearing yoga pants, tights, and it burned right through. So make sure you're always standing, wearing appropriate attire, and you're paying attention to what you're doing. So, again, dip in the isopropyl alcohol, flame so it's sterile. I've been talking and moving around, so I'm doing this again. Once it's sterile, you are ready to go. So, this is where dexterity kind of comes in play. So hold your spreader here in one hand, okay? Get your B tube, and then also get that sterile pipette out of the bag. Set the baggie aside, you're done with it, don't let it get near the flame. And now you're going to decap the tube B, flame it, reach in with your transfer pipette, suck up just a little bit of fluid, you don't need a ton, okay? Flame the tube, cap, set the tube aside, you're done. Now your hands are freed up a little bit. You're done with the tubes altogether, move them to the side. Now you're on your plate. Take that liquid that you pulled up into your transfer pipette and drop it right in the middle of the plate. You only need 1-2 drops at most. Okay? And simply dispose of the transfer pipette in your waste container. Now, with that inoculation loop, now we've been moving a little bit here. Make sure you move quickly because it's only going to stay sterile so long with all this moving around. Take that inoculation loop and simply brush it across the plate here, moving the plate in circles so that you spread that culture all the way around. And while you're doing this, you're not doing like I'm doing and talking, because as you talk, you're going to potentially speak into your dish and contaminate and ruin the whole purpose of this experiment, which is to isolate individual bacterial cultures. Once you've spread that liquid around, it's going to take a moment or two, a few minutes for it to soak into the media. So while you wait on that, go ahead and sterilize. Do a quick pass over the flame just so you're not potentially adding any excess liquid back to your isopropyl alcohol. Dip into the isopropyl alcohol and flame to sterilize again. Again, being careful and mindful about where you're at with this flaming alcohol. This is going to take some time to cool off, so again, just sit here and wait before you just set it down. You're done with the alcohol, so you can cover it with a glass plate. You now have your spread plate finished, hopefully already labeled. And once this cools off, you're good to go.